All right, so we will get started. Okay, so ladies and gentlemen, thank you so much for coming to the Osaka Finance Forum Life Science and Healthcare Startup Pitch today. For today's event, we've invited the 10 startups from Osaka and Kansai region in life science and healthcare industries. That industry is one of the industry that Osaka and the Kansai area has an allure and specialty. So today's event, along with the Global Financial City Osaka vision, we would like to connect those startups with the VCs and other investors in the overseas and those from Japan. And so we are having the pitches of those 10 companies along with the networking at the end of this event. So again, thank you very much for your attendance. So here's today's agenda. So we would start by the greetings from the organizer, followed by the keynote speech talking about the allure of the startup ecosystem of life sciences and healthcare in Osaka and Kansai. Then we would move on to the 10 companies pitch. So we are about to continue this by 7 p.m., then followed by an hour networking till 8 p.m. if you're joining at the venue. So please kindly enjoy your event. Right, here's some introductions and instructions for this event. First, if you're joining from the venue, you are now at the pitch areas, and there's each startups are having the booth at the end of this event venue. So you can visit those company whenever you want. So during the pitches or during the breaks and networking, please kindly visit the booth of individual companies and give some questions or exchange business cards, contacts, or have some meetings with them, please. And also the badges that you are having differs by the nature of each participant. So here's the instruction of the colors indicates each nature, and it is also written on the handouts which was distributed at the entrance as well. Next, for this event, we are having a simultaneous translation. And even though you are joining from the venue, please kindly use the function of the Zoom to listen to the voices, translated voices. So please use your smartphones or whatever if you are joining from the venue but would like to listen to the translation. Also, next, we are having the Q&A session after each pitches. So for each company, we'll have 7 minutes for pitch, then followed by the 3 minutes Q&A. And if you're joining from the venue, please kindly raise your hand, and the secretariat would take a question. And if you're joining online, please use the Q&A function of the Zoom in order to raise your question. And the secretariat and MC would take your question. Next, we are having the materials on the Google Drive. In particular, since on the screen there's only single language, the speaker's language, the material would be there. So if you would like to see the for example, translated material, then please kindly access this Google Drive and have a look on the materials. You may find the link on the Zoom chat box later on, or it is written on the handouts as well. And if you're leaving this event, please kindly answer the survey. And this survey includes the function to raise your interest for the individual business meetings afterward this event. So please kindly access this and also apply for the individual meetings if you want. Right, we would now move on to the keynote speech, which is talked by Ms. Kazuyo Wada about the allure, the attractiveness of the Osaka and Kansai's startup ecosystem and its life sciences and healthcare startups. So the greetings, the keynote speech is from Head of Intellectual Property Strategy Office, Department of Industrial Academic Collaboration, Open Innovation Center, National Cerebral and Cardiovascular Center, Ms. Kazuyo Wada. Right, Ms. Wada, please begin your keynote speech. Thank you.
Thank you for kind warm introduction, Moriyama-san. And to everyone who has joined us here today, whether in person or virtually.
Welcome. My name is Kazuyo Wada, currently heading the Intellectual Property Strategy Office at the National Cerebral and Cardiovascular Center, NCVC. It's a pleasure to be here, and I'm excited to share my thoughts on how important venture capitals affect startup strategic growth.
Here's what I am talking about today. First, I'll introduce the concept and approach of open innovation at NCVC. Then, I'll explore the unique characteristics of life science and healthcare startups emerging from academia. Finally, we'll look at the key roles that venture capitalists play in driving with the innovation ecosystem. NCVC is a leading medical institution specialized in cerebral and cardiovascular diseases. It's also one of 6 national centers for advanced and specialized medical care in Japan. NCVC is organized into 3 divisions: the hospital, the research institute, and the Open Innovation Center. These 3 divisions work together as one integrated organization. From now on, I will introduce our initiatives for open innovation. Our Open Innovation Center includes unique resources like these, which provide startups and collaborators with cutting-edge tools and infrastructure for research and innovation. In the Open Innovation Lab, OIL, companies, universities, and other external organizations partner with us can conduct joint research across a wide range of fields related to health maintenance and promotion. The Open Innovation Center Wet Laboratory, called OR, is quite unique and rare in academia. Our OR enables startups to conduct biochemical experiments without needing their own setup. And Science Cafe is a space for casual, yet impactful discussion between researchers, doctors from NCVC, and companies. And we have an excellent, amazing platform, which plays a key role in cerebral and cardiovascular research. It's also fully equipped with some of the latest technology, including cutting-edge microscopes for use for imaging. And Joint Research Division have been established to promote collaborative research projects with companies more effectively and efficiently. These projects are set for a minimum of 3 years and can be updated for up to 10 years. Joint Research Division reaches industry and NCVC by securing research funding from companies and liberating clinical knowledge from the NCVC. It delivers research outcomes that support commercialization for industry, while also providing results that enhance medical care at the NCVC. There are several key benefits to this system for accelerating innovation, as I talked about in the previous slide. These 4 shown here are ventures established by NCVC scientists that align with the philosophy of NCVC written here. Certified ventures can pay NCVC licensing fee, not in cash but through stock acquisition right, and they are also eligible for deferred payment of laboratory rental fees. They have already delivered remarkable results. From here, I'd like to move to a topic about academic startups in the life sciences and healthcare fields. But before that, normally, most of speakers begin with a self-introduction, but let me start here by showing my career. What you see here is a typical process of pharmaceutical development. What do you see? Oh, okay. After graduating from Kyoto University, Faculty of Pharmaceutical Sciences with master degree, I began my career as a researcher at the global pharmaceutical company Ciba-Geigy, now known as Novartis. I won't bore you with the details as time is short, but I have been able to gain so much experience, and then I am in my current position. I believe that it is impossible to have such a career by working at just one company. Based on these experiments, I will share my insight. Academic IP has these strengths. However, there are also challenges. Academic IP often leads to breakthrough innovations, but requires significant resources for patent management and commercialization. Those who manage to overcome these hurdles are launching startups, I think. Still, big challenge remains the significant cost of patent application and maintaining those rights. Here are some of the expectations and considerations for life science and healthcare startups from academia. On the positive side, the starting point is the desire of doctors and others in the clinical field to cure patients in front of them, which reflect significant unmet medical needs. Because seeds and candidate products are created through the alignment of research and clinical development, their stories are compelling, grounded in scientific rationality. This is what connects academia-based startups to their mission of addressing social challenges. However, from a corporate standpoint, there are some hurdles. Pharmaceutical companies may hesitate to partner with these startups due to uncertainties and insecurities. These arise from concerns about the reliability and robustness of data, insufficient analysis of target of mechanism, or issues with optimization such as inappropriate narrowing of focus. Balancing these expectations and challenges is critical for success. The Kansai is home to a vibrant academic community. Kyoto University, my alma mater, and Osaka University, which is closely connected to NCVC, and Kobe University, where I used to work, have all excelled in blending literatures and sciences, focusing on entrepreneurship education, and fostering many startups. In fact, the two startups I've been involved with originated from Osaka University and Kobe University. Kansai's weakness is just that it's not Japan's capital, nor the center of politics and economics in Japan. In Kansai, local government, universities, and the private sector are working together to create ecosystem in which startups are born and grow one after another, centered on Kansai area. From a global perspective, Kansai's academic and entrepreneurial potential represent a vital part of Japan's innovation landscape. I hope international investors will look toward Western Japan to discover and nurture its incredible seeds of innovation. When it comes to networking, Kansai's culture, rooted in comedy and love for making people smile, makes us naturally open in interacting with people from around the world. Finally, I'd like to highlight the crucial role of aligning startups with venture capitalists to build a thriving and effective ecosystem. The challenges in the management resources of startups are shown here. As to tangible resources often include human resources, physical assets, and funding. Not enough people, not enough goods, not enough money. So, intangible resources, the information they need is not captured. It is time-consuming, and there are also issues regarding intellectual property that I have just conveyed. Without solutions to these challenges, any effective innovation ecosystem like US cannot be established. Actually, I had the opportunity to collaborate with venture capitalists in my current position, which allowed me to recognize the critical role they play. This has been a profound revelation. That is why I strongly encourage those of you in academia and startups to consider partnering with VCs.
VCs can assist with the necessary funding and in some cases help secure public budget. I also encourage you to leverage their knowledge and experience in areas such as these written here. Many VCs possess strong science background and industry experience, making them invaluable partners from early stage strategy and scaling. They can also open doors to valuable networking opportunities. If utilized effectively, it can serve as the most significant catalyst for your success. I hope that today's forum will be one of those catalysts.
For your success. Yes, that's all. And thank you very much for your attention. If you have any questions or would like to learn more about NCVC's Open Innovation Initiatives or co-creation platforms introduced today, please feel free to reach out. Thank you.
Thank you very much, Ms. Wada. All right, so now we would like to move on to the pitch sessions. As described earlier, today we have 10 startup companies from Osaka and Kansai region in life sciences and healthcare startup industries. So from now on, each company would have 7 minutes to pitch, followed by the 3 minutes Q&A. So if you have questions, please raise your hand if you're joining from the venue, or either raise your question throughout the Zoom's Q&A function if you're joining online. And you may raise your question by Japanese or English, either the language is fine. So before the break, we have 4 companies to go first with a short break, and then the 6 companies will be followed. All right, for the first company, we would like to invite Mr. Dan Takeno from ACT. Right, now we are preparing the slides, and yes, now the slides are ready. So Mr. Takeno, the floor is yours.
Hello, my name is Dan Takeno, CEO of ACT Incorporated. We have created the new health index. We are a deep tech healthcare startup. Our mission is looking after the health of people around the world. Cancer, glaucoma, diabetes, dementia, these diseases continue to increase. All diseases are related to blood vessel. Vascular diagnosis and tests in DICE have not been established and disseminated. Do you live too longer? Do you want to prevent aging and your brain and bone from deteriorating? Those are related to the capillary. We have succeeded in visualizing disease by observing capillary with Osaka University. Our product is capillary scope software, cloud-based analysis and application on smartphone. Please watch the video.
Capillary analysis system is comprised from a set of patented technologies for analyzing capillary function. The red moving parts in the monitor are capillaries. The test is non-invasive and doesn't require any kind of license. The soft output measurement of the number of capillaries, length, width, and fuzziness is to identify capillary function. The soft output evaluation of capillary length is a comparison between capillaries and automatically identifies capillary function. Capillary function is strongly correlated to individual health conditions, and capillary shape can change dramatically. If the shape is long and the total number of capillaries is observed to be large, it indicates healthy capillary function. On the other hand, if they are short, fuzzy, or bold, it is a sign of a pre-symptomatic condition. Please feel free to contact us for more information about our technology and products.
Health management market in Japan, $5 billion US. We plan to get 1% of it. I mean, 50 million US. The market is the world is 10 times as much as than in Japan. We sold about 1,800 units to pharmacy, etc. And the number of joint research with institutions is more than 50. An academic paper was accepted as a result of joint research with Tohoku University. Simple approach to detecting and assessing diabetic retinopathy. No invasive nail fold capillary measurement found to be effective. Capillary scope gave visual information on the efficacy of your taking countermeasure. Those examples show the countermeasure are validated. I have been research capillary image 10 years, and I am invited project researcher at RIKEN, a national research institute in Japan. CTO Kaneko is a special engineer for image processing. He registered 100 patents. Professor Takakura is the authority on capillary research in the world and medical doctor. Competitor is a know how to analyze capillaries of 15,000 people at least. Acquire 3 patents for capillary image processing. The only one in the world capable of exacting and quantifying 36 items in 4 seconds. Dataset acquired with RIKEN at 400 items and more than 1,000 people. Competitor is manufacturer of blood composition monitor. The feature of our product are easy to understand, high impact and effective health counseling in a short time by visual and numerical value of the video. In the future, we will plan to develop blood test without drawing blood. The business model is a hardware sales of $7,000 US and a cloud-based analysis service of $100 US per month. There are 50,000 pharmacies in Japan, of which we plan to take a 20% share. There are also a big market for fitness and aesthetic. We have decided to exhibit at the World Expo in 2025, planning an IPO in 2027, and are currently raising 200 million yen in Series A funding. Our mission is looking after the health of people around the world. Now we are developing a brand new capillary scope, autofocus system. Easily take a better focus and wider size image. We will create evidence-based healthcare and expand to the world. Thank you for your attention.
Thank you very much, Mr. Takeno. All right, from here, we would like to take a few questions within these 3 minutes. If you have any question, yeah, all right. Here's the first question.
すみません、こんにちは。B ヘルスの遠藤と申します。
Hello, I'm Endo from V
海
Health.
外展開っていうのは検討されております。
Are you considering expanding your business to overseas? And if you consider it,
国や地域っていうのは
do you have any specific country or area as a target?
海外展開、現行のバージョンですでに問い合わせのあるところ
With
は
the
対応させ
current
ていただいております。
versions, we support overseas Hong Kong, Taiwan, Singapore. There are some distributors already. Eventually, we want to expand our business in Asia and United States.
よろしいでしょうか。
Any other questions?
Thank you very much, and this is very interesting stuff. So, are you saying, that you are finding some, new the biomarker, without using some, for instance, some cell analysis, just like a T cell or other things? That's different things you are talking about.
はい、ありがとうございます。そういう意味では、
Yes, thank you.
何らか、侵襲的に何かを細胞を取ってというわけではなく、
Like taking cells invasively, it's not like that. It's non-invasive and we can see it visually. So based on that visual information, in the future we want to deeply dig so that it can be used for the screening of the blood test in the future.
そうですね。どこまでいけるかは、これからの研究開発。
We are not sure how much we can do. It's based on our R&D.
All right, so we may take one more question. では、はい。
大阪大学創薬サイエンス部門の河野と申します。
I'm Kono from University of Osaka. Thank you for sharing your presentation.
自動数値化オートマティックデジタリゼーションに成功されているんですけれども、このパラメータ
Regarding automatic digitalization, regarding this parameter for diabetes and then other capillary related disease, like what are you thinking about using as an evaluation? And what's the timeline?
まず、既存でですね、相関が出たのが、眼科学教室の方で、糖尿病
Well, we've found that there is a correlation for diabetic retinopathy, and we issued a report. And it's been observed. However, it's not calculated, so we provided the calculation. And for some diseases, including lifestyle disease, there are some correlation with a blood test, so we can work together with RIKEM and National Cardiovascular Research Institution and with other researchers. Cardiovascular related disease. So those are the area we would like to start with. Maybe researchers will be interested in. For class 1, we would like to expand it. And we are currently having conversation with PMD, and then yes, we are trying to make steady development. Thank you. Due to time constraints, let me move on to the next page.
We'd like to move on to the next company. All right. Thank you very much, Mr. Takeno, again. All right. So the next speaker is Mr. Tsuji from Immunosens. Right. Now the presentation is ready. Right. Now the presentation is ready.
Thank you for your giving us this opportunity today. My name is Yoshitaka Tsuji from Immunosens. First, this company profile we established in 2018 as a startup from Osaka University. We raised 600 million. We raised 600 million Japanese yen in 2023. The next round of funding is 500 million Japanese yen planned for June next year. So if you feel unwell, you better to go doctor, but the result will come after 3 days. I'm sick now. I'm so busy. Have you ever experienced like this? So POCT is very important. POCT is point of care testing. So, POCT is a solution to this issue, bring rapid and diagnostic result. So this map is POCT tech map. So immunochromatography is very easy operation and very cheap, but low sensitive. Clinical testing equipment is high sensitive, but very bigger and high expense. So our target field is this: low price, small size, convenience, and high sensitive. So we have an advanced immunoassay technology called GLIA, gold-linked electrochemical immunoassay. Not optical signal, immunoreaction to electric signal. So we realized high sensitive and turnkey operative POCT by combination of GLIA and lateral flow test technology. So now this is our prototype. So it's very small as iPhone charger. This is for consumer. For doctor is a display. So our product set the GLIA stick to the GLIA base and other sample. For example, cerebral and fingertip blood urine. So the result display on the screen. On consumer result on the smartphone. So our strength point 3, quick result will come in 10 minutes, cost effective. Market price 50,000 yen and high sensitive. So the application and market size. Sorry. Sorry. One more. Size. Sample size is 2 or 20 microliter. Application and market size. Medical diagnostics, nurse care, telemedicine, healthcare, emergency. Domestic sum, JPY 300 billion yen. So now we try business development status. For medical, so heart failure, etc. And mental health and sports and animal health. So we are looking for investor and joint partners. So we are here. So last. So we'd like to conclude the presentation with the point again that we can transform the future of POCT. Thank you.
All right, thank you very much, Mr. Tsuji. All right, are there any questions from the floor? We may take one or two questions. And at the moment, there is no question coming from the online. Okay, please.
I'm Kawakami from ASA. The information with low quality, like regarding technology to enhance the quality, like how can you get the high quality information from the data with low quality? How do you do that?
我々なぜ精度が高いかというと、
How can you enhance the data quality? Usually, like optical signal is used. For example, for immunochromatography or clinical test equipment, like optical signal is used. However, for our technology, the number of disease is counted, and that's counted. We have the technology to quantify the data using nanotechnology. That's why we can quantify with high quality. So you measure something else different from the information inserted. But here is a stick, and we can add blood or saliva here. And then this is a sensor. So this reacts. And then it can measure.
Are there any questions? If not, then since we are a bit running out of time, so if you have further questions, please visit the booth afterward. Right, so thank you very much, Mr. Tsuji.
Thank you.
And the next speaker is Mr. Jin Nakamura from GMET. This is the fourth pitch, and this is the last pitch for the first half, first part. And after this, we are having a short break before moving on to the rest of the 6 companies. Now we are preparing the slides. ただいまご案内しました通り、こちら4社目の後ですね、少し小休憩を挟みまして、
As we have announced, we'll be taking a short break after this and then move on to the 6th.
So, Mr. Nakamura, please begin.
Okay, hello everyone. Thank you for joining us today. My name is Jin Nakamura, CEO of GMED Incorporated. So today, I'm very excited to introduce our initiative to revolutionize communication and deliver innovative healthcare solution by connecting the brain with computers. Globally, over 4 million people each year face injuries or illnesses that result in severe mobility and communication challenges, significantly affecting their quality of life. Although they have the will to live, they experience strong pain from being unable to communicate with others, even with their family. Some patients, despite their desire to live, they choose to pass away due to this terrible situation. This situation not only impacts the patient themselves, but also places a tremendous emotional and physical burden on their families and caregivers. Unfortunately, current medical technologies offer no solution to this problem. this is where brain-computer interface technology comes in. Our solution enables the operation of electronic devices such as PCs using brain activity. So as you can see here, our research at Osaka University has demonstrated the operation of various devices using these technologies. By utilizing these technologies, even patients in a locked-in state can communicate with others and support their own physical activities. So our key innovation is this compact EEG device, about the size of wristwatch. It works by placing electrode sheet on the brain surface to capture brain waves, while the device itself replaces part of the skull. Brain waves collected by this device are analyzed by AI, enabling the seamless operation of external devices. A few companies in the world are developing implantable BCIs. North American companies such as Neuralink and Synchron place electrode within brain tissue or blood vessels, which involve significant safety risk. Additionally, once implanted, the devices cannot be removed, posing a high psychological barrier for patients. So on the other hand, non-invasive BCI can only acquire brainwave signal of insufficient strength, making practical operation of external devices unfeasible. Our BCI stands out as a one-of-a-kind solution, combining safety, practicality, and scalability. We also hold critical patent and proprietary know-how essential to realize this groundbreaking technology. Our business model is based on selling medical devices and generating license revenue from applications powered by our BCIs. We've estimated the market size using patient surveys and input from clinical experts. In the US alone, annual revenues are projected to exceed $900 million, with potential growth to $1.8 billion as applications expand. In terms of our go-to-market strategy, we've completed research and development and established a manufacturing framework with key US vendors. For clinical trials, our protocol has agreed upon with PMDA, Japanese FDA, and the trial site have already been selected. We are also collaborating with relevant government agencies on guideline creation for regulatory approval. Additionally, discussions with distributors interested in sales have advanced significantly. So these efforts ensure a robust foundation for the successful commercialization of our medical devices. Media and patient interest in our product is growing. With this momentum, we aim to accelerate social implementation and expand globally. We have secured sufficient funding to initiate clinical trials in Japan, but we are seeking additional investors to support our future global expansion. Our company has a talented healthcare team and supported from numerous key opinion leaders worldwide. In the future, BCI will evolve into a new communication tool widely accessible to the elderly and general public. Its deployment as a medical device is just the beginning. So this is just right. We believe that human connection is essential in life. It's not just about being alive, but about being able to communicate with each other, enriching the lives of both patients and those around them. So we aim to create a world where no one has to give up due to physical disability by connecting patients, families, and friends, realizing all human well-beings. Okay, thank you for your attention.
Right, thank you very much, Mr. Nakamura. So we may take a few questions before moving on to the break. Are there any questions from the floor or from the webinar participants? If you may have, please use the Q&A function. From the floor, yeah, we are having one question from the floor. Okay.
Thank you for your great presentation. I'd like to ask about your. I'd like to ask a question regarding safety and durability. Your device requires surgical implantation, but is there any concern regarding its safety and long-term durability?
Thank you for your question, and this is very important. So, the to ensure the examination is as accurate as possible. So, let me explain in Japanese. So, 安全性に関しましては、まず
For safety, we have reduced the risk. In other words, there's this unique implantation, so there is no side effects to this implantation. It's basically the same as heart-related implantations, the medical devices. It's basically the same, and as a supporting evidence from a clinical point of view, this can be implanted within 2 or 3 hours surgery. And the material that we are using, in the past, it has been proven that it is safe to be put into a human body. So we are using such materials. Therefore, we have not observed any additional safety-related risks. Thank you very much. And regarding the tolerability, do you have any evidence about how many years you have used this? So at this point, approximately 5 years, but we believe that it will be able to be used longer. So we need to use a tolerability test and ensure practically how many years it can be used. Thank you very much.
Are there any other questions? If not, all right. So again, thank you very much, Mr. Nakamura. And okay, now we would like to close the first half of the session. Thank you very much. Right, so as instructed, we are now moving on to the short break. Sorry that we cannot take 20 minutes as scheduled, but we would like to start as scheduled at 5:50. So we are having about 10 minutes for break. And then after the 10 minutes break, we would like to resume the latter half, the 6 companies presentation. Please visit the booth over there at the rear end of this venue. If you're joining on site, and join the booth of each company. And if you're leaving this at this moment, please kindly take the survey and leave us the messages or your interest for the meeting. This survey, you can shot the QR code on the screen. And also, if you're joining online, then there's an instruction on the chat box. So now we are now presenting the venue slide, the slide of the venue, and we are now having a booth over there. The 10 companies is having in each company's booth. So please kindly visit the end of this hall. And as mentioned before, each company, each participants, their nature, their industries are distinguished by the color of the name tag here. Either it is the institution or is it the investors or whatsoever. So please kindly have a look on them to identify to whom you would like to speak. Right, so we are restarting the session at 5:50. Thank you very much. で皆様ですね、50分までの休憩の間、ぜひですね、会場奥の方、ブースで各社さん、ご登壇でおられますので、よろしければぜひご覧ください。ブースで各社様へのぜひご質問、お名刺交換、ネットワーキング商談等、このお時間にもお願いいたします。
Your business. Interesting? Growing? Yes, it's open in Osaka. The 5 reasons. Osaka, open in all the ways that are just right for you. The city and people of Osaka have always been known for its love of doing business. Its personality has given birth to many unique products and services. Many companies established in Osaka have grown into the 5 reasons. More about the 5 reasons on our website. Open Osaka.
Osaka Global Finance One-Stop Support Center is located in the Knowledge Capital Collabo office on the 8th floor of Grand Front, Osaka, and opened in December 2021. We provide support for foreign financial companies and foreign investors who are looking to expand into Osaka. Specifically, our experts in finance provide advice and consulting on financial legal systems and licenses. We provide one-stop services not only through the experts at the center, but also through our partner institutions. Osaka aims to become a global city of finance. To achieve this goal, we've set forth 2 visions for Osaka as a global city of finance. The first is to be a global city that develops through finance by attracting the vitality of both Asia and the world. And the second is to be a financial frontrunner city that challenges the world through pioneering initiatives. To realize this vision of a global city of finance, Osaka Prefectural Government and Osaka City are promoting a variety of initiatives. As part of this effort, the Osaka Global Finance One-Stop Support Center was established. In addition, Osaka will host the World Expo in 2025. The theme of the expo is designing a future society where life shines brightly, and it has the potential to create business opportunities and new innovations in a variety of fields. From here, you can find the Osaka Global Finance One-Stop Support Center in detail. The office that houses the center was built in the Umekita district, in front of JR Osaka Station, the largest terminal in Western Japan. This area was developed based on the concept of creating new innovations by connecting outstanding technologies and people in research and development centers around the Kansai region. We opened our center in December 2021. We will provide free consulting services on financial legal systems, licenses, etc. for foreign financial companies and foreign investors who are looking to expand into Osaka. We provide meaningful support to foreign financial companies and others looking to expand their business in Osaka. There are various barriers for companies and investors to enter the Japanese market, such as Japan's unique legal system and procedures. Our English-speaking financial consultants provide a one-stop shop for 3 main services, and free of charge. First, regarding the provision of information on the Japanese legal system and procedures. When entering the Japanese market, it is necessary to understand the financial instruments and exchange law, the fund settlement law, and other financial laws. And our consultants are able to provide accurate information at any
All
time.
right, this is an announcement. So we are restarting the seminar in about 2 minutes. In about 2 minutes, we are restarting. So please kindly go back to the seat if you're ready. Thank you very much.
FSA on procedures for obtaining financial licenses and can also refer you to attorneys specializing in finance. Support visas, registration, recruitment assistance, and business matching assistance will be provided in cooperation with OBIC, an organization that assists non-financial overseas companies in setting up operations in Osaka. Lastly, we provide other support systems beyond business development processes. We also provide consultation on housing, medical care, education, and other lifestyle-related issues in cooperation with related organizations. We help resolving your concerns, such as life necessities, upon expanding into Osaka. If your company is interested in doing business
繰り返しです。もうあと、間もなくですね、セミナー再開いたしますので、順次お席への戻
We
りをお
will
願いいたし
resume
ま
the
す。
session shortly, so please come back to your seat. Thank you.
Yeah, sorry for keep you waited. We are about to start the latter half of the session. Thank you very much. All right, thank you very much. So now we would like to restart the latter half of the session. Right, here is the instruction again. So we would like to give you the instruction again. All right, again, so from now on, we are having the 6 companies from number 5 to number 10 is given a pitch up until 7 p.m., then followed by an hour networking. All right, again, here's an instruction. Okay, so we are having the simultaneous translation as mentioned. So please kindly use that. And if you're joining from the venue, please kindly use your mobile phone to connect to the Zoom feature. And if you don't have an earphone or have trouble with the devices, please kindly contact the secretary so they will give you some assistance. Next, again, we are accepting the Q&A by the end of each presentation, about 3 minutes. So if you have any question, please raise your hand if you're joining on site, or kindly use the Q&A function of the Zoom if you're coming online. Also, we're distributing the material, so please kindly access the Google Drive. You may find the link here at the screen, or either you may have find it on the handout. So please refer to them. Lastly, we are having a survey, so please kindly cooperate answering them when you are leaving. All right, so now we are restarting the second part. All right, the next company is number 5, PaMeLa. So we are welcoming Mr. Hiroaki Goto to the stage. Okay, now the material is ready. So Mr. Goto, please begin your presentation. Thank you.
Hello, everyone. Thank you very much for this opportunity to introduce PaMeLa today. I'm Hiroaki Goto, CEO of PaMeLa. We are developing AI to quantify pain and aiming to bring objectivity to human pain assessment. The social issue is that pain causes significant quality of life decline and huge economic loss. Even though Japanese pain medication market is significant, 23 million Japanese people are suffering from chronic pain, and 86% of these people has been suffering for years, and economic loss caused by this pain is estimated 2 trillion yen annually. So it's a huge economic loss. Why pain cannot be well treated even though there is a significant market? The answer is that existing pain assessment method is inaccurate because it's subjective and time-consuming. Doctors use VAS for their pain assessment, asking the patient that, how much pain are you feeling with this 10 centimeter scale? The right side means the worst pain in your life, and the left side means no pain. So it's quite challenging for patients to express their pain accurately. And also, doctors are difficult to find sufficient time for their interview pain assessment. A paper reported that 39% of post-surgery patients experienced severe or moderate pain even after the pain management by the doctor. Insufficient post-surgery pain management increases the risk of complications. So this leads to a lower hospital revenue due to the lower bed turnover and additional medication cost. And also, poor pain management causes chronic pain, with 20% still having the pain after 3 months. And only 36% of chronic pain patients answered that they are satisfied with their doctor's care. So this means there is a strong and urgent need to improve pain management and pain assessment. So we are developing AI, EEG-based AI to quantify pain. Electrodes read EEG signals, and which are digitally converted by AD converter and analyzed by our AI. And our PS pain score is displayed as a number and time series data, PS trend data. Our real-time technology can provide 0 to 100 pain score, supporting doctors' decision-making and pain management as the clinical settings. Furthermore, unlike traditional pain assessment method, our technology enables continuous pain monitoring, so medical staff can check patient's pain anytime. We have already confirmed our technology efficacy in human trial. Please see the right side figure. This is the data of human pain before and after the administration of painkiller after surgery. Our pain score measures the statistically significant decrease of pain and showed some correlation with VAS, indicating that our technology can assess human pain effectively and objectively. We've already successfully completed our first clinical trial with Japanese national universities and won Best Presentation Award at a major Japanese academic conference. The feedbacks from doctors were positive. And now we have been conducting second clinical trial with 2 Japanese national universities for the approval. This is a multi-site clinical trial. As for the competitors, there are 4 companies, there are 4 competitors aiming to assess human pain objectively. And 3 companies are using peripheral data, such as heart rate and blood pressure. And only PainQX uses EEG data, brainwave data. And only PaMeLa is a company that has succeeded in clinical trial using EEG. EEG is expected to be more accurate than peripheral data because brainwave data directly reflects human pain information. And the market size is expected to be grow substantially as global population aging progresses. Our strength lies in both higher quality training data and for algorithm and cutting-edge algorithm developed through our long history of R&D, about 9 years. We are raising about $2 to $3 million US now to conduct a clinical trial to compare our technology against conventional ones. This clinical study will validate our technology of clinical merit and operational efficiency improvements by our technology, like patient throughput and bed turnover of the hospital. Because we use EEG data for pain estimation, we believe our technology can change the pain assessment dramatically from post-surgery and chronic pain to cancer pain, sports medication, pediatrics, and drug development. The next clinical trial will be the key for us. If you are interested in us, please contact me anytime and please visit our booth there. Thank you very much.
Right. Thank you very much, Mr. Okay. Sorry, Mr. Goto. And okay, so we are taking the questions from now on. If you may have anyone raising the question. Right. One question from the on-site venue.
Regarding chronic pain, you mentioned that the pain can be assessed objectively. So chronic pain is usually patient get used to it. As a result, like how can you say that objective observation can be done or additional research will be required? Well, to be more specific, clinical trial is required. However, based on a small number of experiments we conducted, we already confirmed. Regarding chronic pains, for example, it includes the mental disease, so it may have impact on EEG. So we need to validate if our algorithm works. However, we already validated that our technology can be applied to the chronic disease based on a small number of experiments. Thank you. For example, patient does not have pain. However, maybe EEG shows the pain result. Does it happen? Well, that's something not observed yet based on our algorithm. Patient needs to feel pain to reflect that on EEG.
If not, okay, then we would like to move on to the next presentation. Thank you very much, Mr. Goto. And all right, so next we are moving on to company number 6, Vision Care. The presenter is Miss Masayo Takahashi. Okay, now we are switching the screen, so please wait for a while. Okay, now it's ready. So Ms. Takahashi, please begin your presentation.
Okay, thank you, thank you. I'm the president of the VC cell therapy. So I would like to present this session. And our Vision Care Group is a part of the Kobe Eye Center initiative that consists to include the Kobe City Eye Hospital and the NPO for the patient care. And our company has 3 companies, Vision Care and the 2 subsidiary companies called VC Gene Therapy and VC Cell Therapy. So in VC Gene Therapy, we are developing the first-in-class gene editing treatment for the retinal diseases. But today I would like to focus on the cell therapy, that developing the cell therapy for the retina. And we have 2 cell types. Retinal pigment epithelial cells, RPE, and the photoreceptor organoid, PRO. Those 2 types of cells situated in the outer part of the retina. And the photoreceptor first perceive the light, and the RPE maintain the photoreceptor. So these 2 are very important cells, and they never regenerate. So once they are degenerate, missing out, we should transplant them after the disease. And good thing is we have this ophthalmology test called OCT. So we can see evaluate the patient's retina in this resolution, 7-micron resolution. So we can tell if the photoreceptor is there or missing or degenerating. Clearly we can tell. And the only 2 types of cells, but that cause the most of the visual impairment cases in the world. And you can see the anti-VEGF market is targeted only 1/10 of the age-related macular degeneration. So our target is 10 times more patients compared to the anti-VEGF treatment. And we have RPE and the photoreceptor organoid, and also the combined cells, because both many diseases cause the both cell type degeneration after severe stage. So we have both, and also we have the in vivo regenerative regeneration of the retinal cells, but this is a still basic research phase. So today the time is limited, so I'd like to just say why you should choose VCCT, the advantage of our company. So we have the numerous human first achievements. Our team did this in successful manner. We are the team that came from the RIKEN laboratory, and we did the first retinal cells differentiation from primate ES or iPS cells. And we proved that we can use those pluripotent stem cells for the treatment for the first time. So science level is okay, RIKEN level, but also we did the project successfully. So we did the world's first iPS-derived cell transplantation in human in 2014. And I was chosen as a 10 people who mattered in that year. And the results are this. Only 1 patient, but we saw the good survival of the retinal pigment epithelial cell sheet transplantation, even after 7 years and almost 10 years now. And you can see the photoreceptor layers maintained just above the retinal RPE cell sheet. And from here, the photoreceptor completely gone without RPE. And her visual acuity maintained without any injection of a VEGF treatment, although she got the 15 times anti-VEGF treatment beforehand, but her visual acuity dropped down, so she could not quit that. But after the treatment, she could quit the injection of the drugs, so she's very happy now. So only one case, but we think we proved the RP sheet long-term survival. That means those cells are necessary in the tissue. And also the maintenance of photoreceptor layer and the choroidal thickness beneath the grafted sheet was maintained. So that means the RPE sheet function after 7 years. So things are happening that would not have happened without the transplant. Also, we did the world's first clinical cells manufactured by AI robot. And with this booth, we actually utilize these cells to the human in 2022. Okay, so we did very many first things, but also already we have the best treatment in the world, we think, because we have the longest history of clinical trial and error. And after the first case, that was the symbolic treatment because at that time, everyone in the world thought iPS cell was a very dangerous cell. So we should prove that it's safe if we utilize like this. And after that, we start developing the standard treatment. So the first case was autologous treatment, but we moved to the allogeneic treatment. And the cell suspension we tried, but we realized the cell suspension, we cannot control the cell's location. So we invented the RPE strip that can be, we can put the cells in the right area that we target. Okay. So now we have this RP cell strip. With this, we experienced all these formulation, but we found out that RP cell strip can be transplanted safely from a small hole. And after the transplantation, they expand as a monolayer, like a sheet. So I think this is the most safe and the most effective formulation of the RP cell transplantation. And actually, the other competitors in the world reported the first results of the first phase 1-2 clinical trial in United States. But compared to that result, our method is better than their treatment. And also we have the more improved photoreceptor organized treatment. Okay. But we have not only the cell products, but we are developing whole treatment as a package. And I myself was a retinal surgeon, so I know how the surgical treatment developed. I experienced with the cataract surgery development, and the new surgery goes very slowly at the beginning, but suddenly it improved dramatically when the many experts involved. So we would like to make this happen as soon as possible, because the patient is waiting. When will it happen? Usually it happens after the clinical trial approval. I know that after the approval, many experts, in this case, retinal surgeons, will try and error and discuss in the conference of the ophthalmology 10 years. And then the treatment will become a standard treatment. But we and ophthalmologists, many ophthalmologists think these cells already work. So I don't want to do the clinical trial only, but we would like to try this sooner alongside with the clinical trial. So in Japan, we have a very good system for the regenerative medicine, and we utilize maximum of it. And we are now applying to the advanced therapy in Japan. So with this advanced therapy, we can accumulate the data and also with fee, so we can earn before the approval of the clinical trial. And the many ophthalmologists think this is reasonable and it can be acceptable for the retinal surgeon. So we made a package because the selection of the patient is the most important part. So we made a register system beneath the Society of Ophthalmology, and we made the explanation software using the generative AI. So now we can transport this package, full package, and the know-how of the treatment to the world. So beforehand, we are trying to make the inbound treatment, and we made a consortium of the retinal surgeon with more than 20 universities. And now we are talking with the Asian people who can get join, and the Singapore and Thailand and Vietnam hospitals are trying to join. So I think this can be a game changer in the medical system, and we can make it sooner and cheaper, I think. So thank you very much for your
Thank you very
attention.
much, Ms. Takahashi. Right, again, we would like to take a question, either it is from the floor or from online. Any question? The secretariat would take the question. All right, gentlemen from the rear, please.
So I would like to ask in Japanese, so how much clinical trials have you conducted and how many cases are you planning to accumulate? So for the clinical trial, we believe that it should be done by the NHA knockout cell. So we are currently preparing for that. And while preparing for that, we would like to do a clinical trial research and identify how to verify the efficacy so that we will be able to be very successful when we actually do the clinical trial.
All right, one question, one more. Thank you very much.
Before going to the market, you have already 3 companies, and could you tell you the reason why you have already 3 companies?
Yes, we will modify the structure for the after the funding, or if we see people wants to modify the structure, we can modify that. But beforehand, we make the vision care first to make those establish the eye center initiative, whole initiative, so that we see vision care, see all the field of the hospital and the NPO and everything. And after that, we made the gene therapy company and the cell therapy company. That's the structure.
It is more efficient to
yes, because we think we made the ecosystem to develop such kind of new modality, the clinical team and the patient including, and the company. So I think this is a very effective ecosystem. Thank you.
Thank you very much. Any other question? All right, then we would like to move on to the next presentation. So thank you very much, Ms. Takahashi again. All right, the next is company number 7, COGNANO, and the speaker is Mr. Akihiro Imura. And sorry, we are just preparing the slides. One moment, please. Okay, then now the presentation is ready. Thank you very much. The floor is yours.
Hello, everybody. I'm Akihiro Imura from COGNANO, a tech bio for new AI drug discovery. Before my presentation, let me share a very interesting movie, Moneyball. Moneyball is very interesting movie focusing on the importance of statistics and mathematics in business. The manager of weak team consider what is the correct way to win. So the old style repetitors and consultants are recommended power no shoulder or a strong power and batting and home runs. No, he rejected the opinion and he hired a mathematician from Yale University, and he employed statistics. And as a result, they won 20 serial games. That was a record, world record in history. Thank you for patience. I mean, we have to say goodbye for old bio and old medicine. I want to go for new biology based on big data, statistics, and algorithm. Okay, here is my team. COGNANO composed of 3 bio experts and 3 IT engineers, and we are collaborating with Google and AI top consortium named OpenFold, as well as global universities. Here is market size of oncology, and within 8 years, the scale amount will reach 600 billion. Let me remind the original story of biodrug. 30 years ago, Dr. Schreiman focused on the target molecule named HER2. The drug name was Herceptin. That was approved 1998. This is the beginning of biodrug history. However, for 30 years, we have no new HER2 still now. So we have no apparent target like this. This is the largest pain in pharmaceuticals, in my thoughts. So there are so many candidates of target molecules for cancer drugs, such as HER2, TROP2, or Claudins. However, they are also expressed on the normal cells. This is the cause of side effect. Okay, in general, on the cell surface, there are so many molecules. So, which is which? This is which is the appropriate target, next target. So, nobody knows. So, we decided to dive deeper. We develop a new technology of digital information of structure. This is nanoscale, and next, Angstrom scale. This is the surface of molecule. There are so many structures, but nobody knows the structure. However, we developed a digital probe of structure using the VHH antibody technology. This is a key or probe for subtle structures with digital label. VHH antibody is a small compound from camelid animals, and this is a very useful tool and easy to be read by next generation sequencer. Our bio team accumulate day by day the big data of antibody coding genes from our buckets. And IT team analyze which is the new drug which is the hopeful novel target. So the principle is very simple. The protein and antibodies are written in alphabetical letters because they compose of amino acid residues. So natural sentences seem like human sentences. So they are corresponding to each other. So fortunately, they are understood by large language model like ChatGPT. Yeah, gotcha. So we collaborate with Google and new OpenFold, and this achievement was accepted by top AI conference, New Rips. And further, of course, we wanted to confirm the efficacy in animal model. So we eventually found new biomarkers on TNBC, triple negative breast cancers and pancreatic cancers, and we made in-house drug, ADC drug, using these antibodies, and we tried to inject in mice, and the evaluation was 2,000-fold efficacy in comparison with ordinary drug. So we are launching serial ADC drug and novel, as well as novel biomarkers every half year. Repeatedly, we are aiming disruptive innovations, and our product is not only drug, but also novel target. And we hope, in our hope, ROI should be more than 30 times. This is our business model. We are collaborating with IT techs and universities in the world, and based on our patents, we a collaborating team launch product as well as joint ventures that would be supported by government. And within 3 years, JV will get exit. And eventually we got a big return from the exit, and we will provide a big return for investors. Okay, this is final slide. Today, everybody can get access to any destination in the world. Similarly, every pharmaceuticals can get any novel targets on the cells or on the molecules with COGNANO map. Please feel free to ask and join COGNANO. And we are looking forward to hearing any interest from you. Thank you so much.
Thank you very much, Mr. Imura. Okay, so now we are moving on to the question. If any company available? Anyone from the floor? And if you're joining online, please use the Q&A function if you have any. Anyone would like to raise question? Okay, from gentleman from the rear. Thank you very much.
Let me speak in English, Japanese. Thank you for your presentation. So the target and antibody, so they are matching. I think you had the slide. Could you show? Oh, yes, this is a slide. So could you elaborate this slide? Yeah, sorry, I just provide a very simple idea only. So basically, antibody and the antigen are matching one by one. And there are a lot of antibodies and antigens as well. So which ones are corresponding? So that is... very difficult to figure out. So that's why we use big data as machine learning sequence. Well, based on the sequence, we can find out the correspondence. In addition to that, cancer cell and normal cell, when we compare them, there are some antibodies which have affinity with cancer cells, and we can extract it. So we invented this technology. But it's a bit complicated concept. So if you are interested in, I can provide more detailed information.
Thank you very much. As Mr. Imura mentioned that we are having plenty of the time for the networking. So during the networking, please visit the booth and have more deeper discussions. All right, since we are running out of the time, we would like to move on to the next company. So thank you very much again, Mr. Imura. And the next speaker is he for you. The speaker is Mr. Kohei Kishi. And please wait for a while. We are preparing the slides. Okay, please begin. Thank you.
Okay, so thank you very much. So I'm very glad to be here to introduce my company, C4U, as well as about technology, CRISPR-Cas3. So some of you are familiar with CRISPR gene editing technology, and that is a CRISPR-Cas9, but our technology is CRISPR-Cas3. So I will explain what is the difference between the Cas3 and Cas9 today. So C4U stands for the CRISPR for you. So I think that you don't remember CRISPR, but please remember C4U. So our company was established in 2018, 6 years ago from Osaka University. So Professor Mashimo, who was in Osaka University, he discovered CRISPR-Cas3 and he established the company C4U. And now he moved to the University of Tokyo so that we have the 2 research laboratories in Osaka University as well as the University of Tokyo. And CEO and President CEO is Akihiro. And he joined the company 4 years ago, and he is not the founder, but he is an expert of the managing the company. And our CFO is Sonoda. He is a CPA holder, and he is also the board member. And DCIP head and Narita-san, and he is also the board member of the C4U. So this slide is very important for C4U. What is the difference between the CRISPR-Cas9 and Cas3? So we have identified at least 3 big differences between them. Number 1, the high selectivity. So as you may know, CRISPR-Cas9 utilizes 20 bases to recognize where to delete in the gene. So that means 4 to the 20th power selectivity. On the other hand, our CRISPR-Cas3 utilizes 27 bases. So 4 to the 7th power more selectivity by using the CRISPR-Cas3. At this moment, we do not have the off-target event. Off-target means the undesired deletion of the gene. The second point is the type of the deletion. So CRISPR-Cas9 can cut the gene like a scissors as a point cut. On the other hand, our CRISPR-Cas3 can delete a larger portion like a shredder. So that is a quite big differences. So if you consider the completely knockout of the gene, our CRISPR-Cas3 would be more appropriate than CRISPR-Cas9. And last but not least, the IP is quite different. That is a different point. So as you may know, CRISPR-Cas9 discovered 2012. At that time, a lot of organizations insisted the ownership of the CRISPR-Cas9. For example, Broad Institute and UC Berkeley, and also the Vilnius University, they are insisted that is ours. On the other hand, our CRISPR-Cas3, as I mentioned, Osaka University discovered this technology, and we received exclusive license from Osaka University. So if you consider the business by using the CRISPR-Cas3, please contact me. And importantly, there is no overlap pattern between the CRISPR-Cas3 and Cas9. It's very completely separated. So it's very good. So considering such kind of profile for the large deletion, these indications will be the appropriate indications. And mucolipidosis, actually, we are developing CRISPR-Cas3 for this indication, and also the CNS repeat disease, and beta thalassemia or the SCD, and also the DMD, also the target indications by using the CRISPR-Cas3. So these are pipeline. So as I mentioned, we have the two business models. One is our in-house pipeline. So we are developing mucolipidosis with the National Center on Child Health and Development in Japan. And currently we are in the discovery stage, and we are planning to the human in 2026. And also our research team is ongoing to develop the second asset. At this moment, we cannot disclose it, but we are seriously discussing our researching activities. And also, we have the several partnerships with a venture company and also the pharmaceutical companies. For example, we have the research collaboration and license agreement with Noiroimmune, and they are the CAR-T company. So they utilize our CRISPR-Cas3 for T cell. So gene-edited T cell is modified or utilized for their CAR-T platform. So that's one example. And we have the several collaboration with Sumitomo and top Japanese pharmaceutical company. So in the Sumitomo case, we had a research collaboration agreement with them. And this April, they got a good result during the research collaboration, and they have exercised the option to enter the next stage as a licensing agreement. And not only the Japanese company, but also we have the Switzerland Hiliba company, that is a venture company for the dermatology area. So both of us are developing epidermolysis bullosa for the very rare diseases. And also we have the several collaboration with the Cerebral or CDMO SiteFact. And yes, so we have received the Series B financing. So last May, 11 million US dollar, 15 億円, 15 Japanese 億円. But we are planning to the next Series C early next year or mid next year. So not only the pharmaceutical area, but also we have the several collaboration with other industries, such as the plant, agriculture, or fish, or microalgae area. So as you may know, so genes are located in any creatures. So not only the human, but also the plant or animal or fish. So thank you very much for your attention.
All right, thank you very much, Mr. Kishi. Okay, Q&A again, if you have any. Okay, gentleman from the second row from the last. Okay.
Thank you for your excellent presentation. I have a question about your in-house pipeline. So why did you choose to target mucolipidosis? And are there any competitors developing similar drugs?
Thank you very much. So as you may be familiar with that disease, so it's a very rare disease. So at first, we would like to establish or show our efficacy in a human as soon as possible. So we have selected such kind of diseases, the short period. So at this moment, it's very rare and the number of the patients is very small. So we will try to expand our technology to the next disease. So at this moment, I do not understand the direct competitor because it's very, very rare disease. Is that okay for your question?
Okay, thank you.
You are welcome.
And one more question from the front. Any other question? Okay, please.
Yeah, Christopher Cas3 technology is very fantastic because no overlap the patent, and but around these technologies, you need to some drug delivery systems. And how about the drug delivery systems patent? How do you handle such problems?
Thank you very much for your question. So at this moment, so for example, mucolipidosis. So we are developing it. So CRISPR-Cas3 for the mucolipidosis by ex vivo setting. So hematopoietic stem cell is extracted from a patient, and outside the body, we conducted a gene editing platform and return to the patient. So that way the extraporation system of conduct. CRISPR-Cas3 is larger than the CRISPR-Cas9. So as you may know, CRISPR-Cas9 utilizes adeno-associated virus, AAV, to carry the CRISPR-Cas9. So that or non-viral delivery systems such as LNP, ribonucleoprotein particle. Yes. So currently, so BD team, including me, is contacting the several companies for the such kind of delivery system. So C4U doesn't have such kind of delivery system by ourselves. You're welcome.
Right, thank you very much. We may move on to the next presentation. So thank you very much, Mr. Kishi, again.
Thank you very much. Thank you very much.
Okay, so we are having two more companies, company number 9 and 10 to close. So now we are moving on to the Revascular Bio, Mr. Kazuo Omori. Okay, now the floor is yours. Please begin.
Hello, everyone. I'm Kazuo Omori, the CEO of Revascular Bio from Osaka University. So we aim to change the patients' lives with true vascular cell therapy. So I'd like to talk about our company's project. At first, one in 5 deaths is still due to heart disease worldwide. The core problem is the vascular system. Many factors such as aging and diabetes gradually and silently damage the blood vessels and eventually leading to the many vascular diseases, including heart disease. Actually, I'm working as an MD and treating these vascular diseases. But in reality, these vascular diseases are very common and the suffering of patients is very enormous. When the blood vessel is damaged, the supply of oxygen and nutrients is impaired and the organs cannot function properly. There are many therapeutic options for the damage of large blood vessels, but a critical challenge in this area is that there's no cure for the damage of micro blood vessels. So this issue is extremely distressing for both patients and healthcare provider. We can change this situation completely with unprecedented vascular cell therapy. Our core technology is world's first discovery of vascular endothelial stem cell, ESC. In fact, blood vessels is always creating in the human body, but the stem cell is responsible for this angiogenesis were unknown. Our stem cell, ESC, is exactly this kind of stem cell responsible for human angiogenesis. And this technology have been patented in Japan and the US as well. So this stem cell enables two therapeutic concepts. First one is microvascular regeneration by ESC transplantation. And second is delivery of different factors. Actually, the function of blood vessels is not only to deliver blood, but also producing many factors such as coagulating factors. For example, this deficiency of coagulating factors causes hemophilia, and this is restored by ESC transplantation. The most distinctive feature of this technology is its innovative direct angiogenetic effect. This green structure shows that new blood vessels generated by just single ESC transplantation. No other technology can prove this kind of unprecedented effect. So why is this technology can be developed to the therapeutic drugs? It's because three breakthrough innovation in ESC. First one is, of course, discovery of ESC. But not only this, we've already established a very simple culture method by using human subcutaneous tissue. And third, we've already established the POC for the treatment of vascular diseases by ESC transplantation. Our product in fast pipeline is human ESC patch, as shown here, with very simple manufacturing method. And we've already demonstrated that human blood vessel is can be created from this ESC patch just by applying this patch to the disease area. The competitive technologies are gene therapy and other cell therapy, but only ESC therapy can have high efficacy even in severe diseases. This is because only ESC can achieve the direct angiogenesis. In other words, ESC is like seed of angiogenesis, whereas the competitors are like fertilizer. We plan the indication strategy as shown here. Firstly, we will establish the safety and POM in digital alpha of SSC, systemic sclerosis, because of deep unmet needs and low risk in first in human trial. And secondly, we will develop the blockbuster drugs in hemophilia because of enormous market and groundbreaking therapy. There are particularly huge market in the US and globally. The annual sales scale in the US will be $600 million in digital alpha of SSC and $2 billion in hemophilia. You can see development roadmap here. We've already raised seed funding last year, $1.5 million. And also we've got the AMED grant, which doubled the amount of VC funding until 2031. And we are now in the preclinical phase in fast pipeline. After establishing manufacturing process and safety assessment, we will conduct phase 1-2 clinical trial from 2026. And for hemophilia, after establishing the preclinical study in 2027, we will conduct global clinical trial. This is our founder team. I'm MD and have been involved in vascular diseases more than 10 years. And Professor Takakura have more than 30 years of experience in the vascular research. And the COO Nishikaku have a wide range of experience in pharmaceutical company and VC. So we definitely make dreams come true through this team with diverse medical background. And we will change the patient's life really. Thank you very much.
Thank you very much, Mr. Omori. Okay, as usual, we are moving to the question. Okay, anyone? We may take a few questions. Okay. Is there anyone? All right, please.
Yeah. If you expect the delivery of deficiency factors, and how do you administrate ESC? How do you give ESC to the patient?
Thank you so much. It's a very good question. Actually, especially in the fast pipeline, we can use autologous transplantation. And in the hemophilia, for example, we want to restore the protein. So we have to use allogenic transplantation. And also in our data, if we transplant this stem cell to the liver, the stem cell can be differentiated to the sinusoid and to produce the coagulating factor. So now we are thinking to transplant this stem cell to the liver. Yeah. Maybe if I could, we want to put the cell patch on the surface of the liver. Actually, we have a data when we put the cell patch, this cell patch can create a coagulating factor. But yeah, we have to think about that more.
Okay, I can understand.
Thank you.
Thank you very much. We may have time to take one more question, if available. If not, okay, then thank you very much, Mr. Omori. And okay, thank you very much. All right, so ladies and gentlemen, thank you so much for joining us. And as Mr. Takubo mentioned, it's 2.5 hours, and thank you so much for coming along with us for the 10 pitches and other speeches. All right, okay, first, just a very brief advertisement about the oncoming events. Oncoming events. Okay, as you may be aware, the tomorrow is the Hack Osaka day, so that actually we plan this event adjacent to that, and there will be many pitching events, talking sessions, or the exhibitions of the startups are taking place in Osaka as well. So please kindly visit it as well. And also, it is a bit far from now, but during early March, we are actually there's a Japan FinTech Week is planned, and along with this global financial city Osaka vision, in coordinates with the central government, there's various events regarding the financial sector or the fintech is taking place during March. So please book your calendar as well. That it is planned that during this week or adjacent weeks, there's an event also planned in Osaka. So we will be glad to announce that once we are ready. So please be prepared for that as well. And there should be further many other events would be taking place in Osaka. So please keep eyes on us. And so we are distributing this information throughout our website or our SNS. And also today's event will be broadcasted throughout YouTube later on. So if you have missed any of the presentation and would like to have a look on them, please kindly visit and please bookmark or please keep eyes on those accounts, each of the accounts and each information. All right, so lastly, again and again, sorry, but please kindly cooperate with answering to the survey, including your inquiry for each individual company, especially if you're joining online. We would be happy to arrange the one-on-one meeting with the 10 pitch companies today. So please kindly access this. You may find it the QR code here, and also our secretariat would put it on the chat box. So please kindly answer this survey and show your interest for the 10 companies. All right, so again, please give us a big applause for the 10 companies pitches, and we would like to close today's Osaka Financial Forum. Thank you very much. For online broadcasting will be come to an end at this point.